Round two. In this video, as a follow-on from our discussion of the Memphis Meats patent, where we went over an overview of how the whole patent works, we're going to discuss the klf4 and oct4 genes and the transcription factors that they pertain to. So, what are those two genes? What are their pathways? Why are they important? What do they do? So, both of those genes have been found to be really important in the regulation of proliferation. They've also been found to be, if you have just those two genes being highly expressed, they're enough to induce pluripotency. So simply by expressing those two genes in a differentiated cell, we're able to take that cell from a differentiated state to a pluripotent state. Now, the ways in which these specific genes work is reasonably in-depth in terms of bio, but we're going to try and talk about that in a little bit of detail. Okay, so I've done up a wee diagram of the signaling pathways through your cell, and we're going to look at how oct4 is induced by the various cell pathways, and then we get a better understanding of how this is important in terms of your growth factors and your hormones. So, FGFR, functional fibroblast growth factor receptor. There's FGFR 1 through 10, and those receive signals from FGFs, functional fibroblast growth factors. Now those go through PI3K, AKT, and they also go through ERK1/2, 1 or 2, and they feed into oct4 or SOX2. SOX2 is another one that we might discuss in a little while. Same goes for Wnt signaling. There's a whole lot of crazy stuff that goes on with that. But that goes through beta-catenin, also producing oct4. Same goes for TGF-beta receptors. Those goes through SMAD2 and 3, also going into oct4. And finally, your BMP4, going through SMAD1, 5 and 8, into oct4 and SOX2. So all these different signaling pathways, which are used to control your cell in many different ways. All these signaling pathways are responsible for a variety of different things. In this context, however, all of these signals, your FGFs, your Wnt, your TGF-betas, and your BMP4s, all feed into oct4 and SOX2. All of them feed into cell renewal. So what I'm saying here is, if we can control the transcription of oct4 and SOX2 in this context, we can control much more directly the transcription of self-renewal genes, self-renewal proteins. Which means that we don't have to go through the FGF route to go through P1-3K, ERK, AKT to get to the oct4, which is the outcome we're aiming for in controlling our cell to be continued, to proliferate. Instead, we genetically modify the cell such that we have a direct signal for oct4. Now, let's remember our direct signal. It is a TET-on system, which involves doxycycline binding on to ART. I think it was RTA. Right? Now those two bind, and they transcribe the oct4, which is the same gene that is controlled by these 4 signaling pathways. So what we're doing with our genetically modified cell is that instead of having to use these 4 complicated and somewhat superfluous cell control routes, we can directly control the oct4 expression. We can directly control the oct4 expression with the induction of your TET-on approach, with the induction of doxycycline binding with RTA to transcribe oct4. To transcribe oct4. In addition, we also transcribe our klf4, which is very similar to oct4 and SOX2. Just remember that in this case, SOX2 is actually not being used. SOX2 is just similarly triggered by all of these pathways, and it has similar behaviors to oct4. But remember that in the Memphis-Meets patent, SOX2 is not mentioned. SOX2 is not mentioned. Instead, we have klf2. We have klf2. So that's how Memphis-Meets is a more efficient approach to cell signaling control, because they've used genetic modification to circumvent a lot of complicated machinery in terms of cell control. In addition, your functional growth factors that you do need to control your cell, as I mentioned in the previous video, are expensive, are prone to breaking down, are difficult to produce. And so instead, if we use the TET-on signaling pathway, which I'll explain in the next video, we can directly transcribe the oct4 gene, and we can see the effects of that in a more direct way. So, in another video, in the next video, we're going to explain what TET-on signaling is, and in the third one, we'll explain what exactly oct4 does.